See, I'm behind anyway too. All right, let's see. I have iClicker to still start. Okay, now I think we're cooking with fire. All right. Okay, so don't forget to check in on iClicker for attendance. Yep, I always try to remind you guys not to forget to do that. Yeah. It only works if you come in on time, right? Otherwise, I hide it. Okay. Go straight. So let's talk. Oh, before we do that, let's talk about the schedule real quick so that we all know what's happening. Where's the schedule? Where's the schedule? Class calendar here. Okay. So we are Tuesday. We are today. What is Thursday? Case study number 1. So if you're presenting, you have to have your presentation, right? And everybody's got to take a turn presenting. So it's not just one person presenting, everybody presents. And then whether it's your presentation or not, doesn't matter. Everybody has to come in with a printed answers to all the questions in case study number 1. And if you are not presenting, what else do you have to have on that printed sheet of paper? Couple of questions. Okay. So one thing when we're doing those, the little talks and stuff, I am not going to be grading you if you ask the question that you wrote down. So like if somebody else asked your question, it's okay. You come up with something else to ask, that's great. So don't feel like, oh my God, I have to get this question in. Sometimes the discussion evolves and you think of something else you want to know, and that's cool. So don't feel like I have to ask this question, but it's good to come in with some ideas. Cool. Questions? Okay. All right. So now we can start. Okay. So we talked, chapter 13 is all about carbs, right? So what do we call carbs now? Glycans, right? So if you have something that is a glycoconjugate, let's scroll down for a second and talk about this real quick. So a glycoconjugate. What does that mean? You have a sugar, G-A-R, sugar plus something, right? And the amount of the sugar and the something can vary, right? So let's talk about, there's kind of 3 really big classes that we'll talk about, and there's some subgroups within those classes. But the first one are glycoproteins. So what do you have in a glycoprotein? Sugar plus proteins, right? Okay, which one is more prevalent? You have more proteins, right? So what happens is, this happens in the ER, right? In the lumen, ER lumen. You have your protein, and then you add to that whatever your glycan group is, right? That's what happens in the ER. And we'll talk a lot about glycoproteins today, but that's not the only kind of glycoconjugate that has to do with proteins. You can also have proteo, O-T-E-O-L-Y-C-A-N-S. Why is it so hot in here? It's terrible. No, it is not. It's not just any of you. I don't know. Maybe if we want to, open the back door, maybe we get a little draft coming through. I can't open the windows, but windows are not openable. No. Okay. So proteoglycans. How are proteoglycans different than glycoproteins? This is protein plus sugar, right? And whatever you end in, that's what you have more of, more sugar, right? Okay, now there's kind of a sub-classification of proteoglycans, and those are peptidoglycans. Peptid, P-I-D-O-G-L-Y-C-A-N-S. This is a whole protein. A peptidoglycan has amino acids, but it's very short. So you have really, really short protein sequences attached to a ton of sugar. So they're like proteoglycans, but the protein part is really, really, really short. Okay. The next set that we're really not going to talk a whole lot about in this chapter are glycolipids. And what we'll actually do is we'll talk about this when we talk about lipids in chapter 15. We'll talk way more about them. But what are you going to have more of? Lipids or sugar? Lipid. You can have more lipid. And so the lipids, the sugars get attached to the lipids in the Golgi. So depending on what you are glycosylating, tells you where that glycosylation is going to happen, right? Okay. So now that we kind of have our definitions, right? Okay. Go down one time.
Yeah.
Okay. All right. So the two that we're going to focus a lot on in this section are glycoproteins, right? So that's what we said. If it's a enzyme protein, most of it is protein. Now these, you have to think about, what do you need glycosylated proteins for? Like, what's the point of having a protein that has sugar moieties attached to it? Some of them you need on the outside of the cell for like cell signaling and stuff like that, right? So what you want to do is you want some of them to be transported. So if you're going to be in a membrane vesicle, right? If you're going to be transported in a membrane vesicle, these are either going to go for insertion in the membrane, or what else might you need it for? You've heard of the extracellular matrix? What's in the extracellular matrix? A lot of structural proteins and a lot of glycoproteins. So, or secretion to ECM. ECM is extracellular matrix. Okay. Or they could be targeted to certain organelles. Some of them are like the mitochondria, perioxysomes, P-E-R-I-O-X. Oh, I cannot spell. P-E-R-O-X-I-S-O-M-E-S. Perioxysomes. So you got to think about what you want and where it's going to go, right? Okay, so proteoglycans. Proteoglycans are similar, except you have more sugar with a little bit of protein, right? These tend to be exclusively in the extracellular matrix. And so when you look at that extracellular matrix, like if you look at any of the joints and you look at all the cushioning things that are located in the extracellular matrix at those joints, like the gel, right, that supports all that movement, that's proteoglycans. You're going to see a lot of that in joints. And they tend to be covalently linked to an aminoglycan. So what does that mean? Right. So it's going to attach to one of the side chains of the amino acid, and that's what's going to link it to the glycogen, to the sugar. Okay. And we'll show you. So I'll show you some linkages and all kind of stuff like that. Okay. So let's talk about proteoglycans, right? So if we're talking about proteoglycans, it's kind of important to remember that right now we're talking about eukaryotes. Because we're going to talk a lot about peptidoglycan, and peptidoglycan is bacteria, whereas proteoglycan is more eukaryotic. So kind of keep that division in your mind, eukaryotes. So there are a lot of different functions of proteoglycans in the cell. Some of the proteoglycans are actually attached to the membrane and some of them are free floating, right? So here, right, we have one protein strand. It's completely in the extracellular matrix. And you have, this is the protein, this is the green, here's the protein. And then each of the individual sugar moieties is what you can see. So that's why when you look at diagrams and things like that, if you're looking at something that is a sugar, you will see the individual sugar moieties. Like they want you to recognize that. It's a monomer, right? A monomer of sugar. And then you can link proteins to different things once they're glycosylated. There's all kinds of different things that can happen. But you can have some, for example, here that have helical portions that go entirely through the membrane and span and go all the way into the cytosol. So you could have propagated effects that occur when this becomes bound, right? Anything that has a transmembrane domain can facilitate signal transduction that'll go through into the cell. But some of them you just have an anchor, right? This is a GPI anchor. And so this is more like, okay, I want to attach to this place or attach to this cell. Not that I want to make anything else happen, but I need an attachment point. And that could be within one host, like cell to cell within one host, or an infectious agent binding to a host.
So if it just has the anchor, it's still considered transmembrane?
No, this completely spans the membrane, but it can be embedded in the membrane. That would be the anchor over here. But regardless, right, if you either have a transmembrane or you have an anchor, then you are defined as a cell surface proteoglycan. Otherwise, if you're free floating, these are all extracellular matrix proteoglycans.
Their cell surface proteoglycan is how like something can be recognized by itself.
Absolutely. Absolutely. Sometimes, a lot of times it's immune interactions, right? How do I know what cell to target, that kind of stuff? Well, what's sitting on the outside of it? We'll talk about the ADO blood groups. That's going to be really important, but all kinds of different ones. I'll give you some examples, but there's so many out there. Okay, so now we're going to switch gears. That was proteoglycans. This is peptidoglycans. So peptidoglycans, we are no longer eukaryotic, we are prokaryotic, right? So peptidoglycans are found in our bacterial cell walls, right? So when you think about a bacterial cell wall, it's not like a eukaryote, right? So it's almost a lot like a plant, right? Where you have that really rigid outer surface, and that is our peptidoglycan cell wall. Okay, so these peptidoglycans have tons of carbs, and then it's like strings of carbs, string after string after string of carbs. But if you just have like spaghetti, one on top of the other, is that very structurally sound? No. What do you need to do? You need to cross-link it. So the way that you cross-link it is through those short protein peptide sequences. And so that's exactly what happens. You have these spaghetti strands, and the spaghetti strands become linked because, actually not because they link with each other. I can't bend my hand that way, but that protein actually interacts with one another, not with the protein that actually interacts like that. I'm gonna show you. But this is why bacteria have particular shapes, right? When you study microbiology and you see all the different cocci and spherical and so on and so on and so on, that's why they can maintain those shapes, because they have rigid cell walls. But it also serves as a physical boundary to keep things in and out, right? So what are those strands made out of? Those are hexosamine polysaccharide chains. I'm going to show you a picture in a minute. But I don't know anybody who's not had to take antibiotics for something at some point in their life, right? The most common antibiotic, which some people have an allergy to, right? So you have to be careful, is penicillin. So penicillin prevents the cell wall from forming. So it doesn't actually kill bacteria. It prevents bacterial cell growth by inhibiting cell wall biosynthesis. So remember how I said you have the spaghetti strands of sugars, and then the proteins that come down and a protein that comes down and they link together? So if you inhibit that linkage, it's no longer stable and the cell wall falls apart. And so that's exactly what penicillin does, is it prevents those oligopeptide linkages. And so now your cell wall's junk, right? Can't form. So does it kill bacteria? No. So what kills the bacteria? If you have a bacterial infection and you're taking penicillin, what actually kills the bacteria? Your immune system. The bacteria just prevent new ones from forming. That's why sometimes if you have a bad infection, it takes a long time for the for the antibiotic to have an effect, right? Because, okay, we're not going to make any more, but you're pretty gosh darn infected. So it takes your body a while to overcome that infection. Like, I don't think people realize that, right? So really sort of interesting. And so my undergraduate degree was in microbiology. So this is why this stuff really makes me excited. Okay, so if you look at a peptidoglycan wall, right? So these squares represent sugar molecules. So what is GLC-NAC? N-acetylglucosamine. So N-acetylglucosamine is attached via an O linkage, right, to MUR-NAC. I always start with the NAC because that's how you're going to say it. So it's N-acetyl muramic acid. So it's just a repeating, right, over and over again, dipeptide, disaccharide. Thank you. I was like, that is not the right word. Disaccharide. Muramic. M-U-R-M-I-C. Really. M-U-R-A-M-I-C.
Okay, thank you.
Sorry, maybe I spelled it wrong the first
No.
time.
I can't.
So if you look, coming off of the N-acetylmuramic acid, we have this really short peptide sequence. Is this a protein? Anybody know a protein that is 3, 4, 5 long? No, not really. No, right? That's a peptide. That's why we call it a peptidoglycan layer, is because it's just a short peptide. So bacteria have an enzyme called transpeptidase. And transpeptidase is going to actually link one chain to the next, and so on and so on and so on, right? And you're going to have all of these cross links happening. And so it's going to be a super stable cell wall, which is why it's very, very rigid. So if you actually zoom in, here's our natural transpeptidase substrate. So in the red, right, this is the binding site for the enzyme. So if we look at this, let's create an analog, right, that mimics the natural substrate. So we have a structural analog that mimics it. We call this square the beta-lactam ring. So if you are allergic to penicillin, you're allergic to anything that has the beta-lactam ring. Okay? So if you do that, now this is going to bind to your transpeptidase and it's going to soak up the enzyme, right? And then your cell wall doesn't form. But what happens sometimes? Sometimes you take a course of penicillin, and is it effective? Most of the time, yes, but sometimes no. So that's what we're going to talk about today.
So if the penicillin family has the beta-lactam ring, Like that's how they classify.
It's structural. It's all structural.
That must be what you're allergic to. You're allergic to penicillin and other foods.
Yep. So it's all about structure driving function, right? So if you're allergic to one, you're kind of allergic to them all. So is it a perfect mirror image of what the natural substrate is? No, we only really care about a really small portion of it, but this really small portion of it perfectly matches our substrate. Okay, so then what happens, right? You've heard of MRSA, right? Methicillin resistant Staphylococcus aureus. If you had asked me, how long's it been now? 2.5 years? 3 since COVID hit, right? If you had asked me a day before COVID hit, what is going to be the next pandemic? This. Maybe not MRSA in particular, but antibiotic resistant bacteria. And I really, before you walk out here today, I want you to understand how dangerous this can be. So bacteria are phenomenally awesome organisms because they have been around since the beginning of time. You know, like they have been here for so long that they are able to evolve and adapt to basically anything we can throw at them. So if you take a bacteria, you give this bacteria penicillin, most of the time, right, it is going to inhibit the transpeptidase, right, because it's going to bind to the transpeptidase and our transpeptidase becomes inactive and we inhibit cell wall synthesis, right? That's the best thing that could happen. But sometimes you can have a penicillin resistant bacteria. This penicillin resistant bacteria contains transpeptidase, but it also contains beta lactamase. What is an ACE? It's an enzyme, right? So if the beta lactam ring is what gives penicillin its functional ability, if you cleave the ring, it's like not having it. So bacteria, and they can get it a couple of different ways. They can evolve it on their own, or they can actually share DNA, right, and pass it on. So there's a couple of different ways that it can happen. But it'll hydrolyze the beta lactam ring, and now there's no effect on transpeptidase, and bacteria keep growing, right? They're happy. So then we said, okay, fine. You want to be resistant to penicillin? We're going to make another drug. And we don't actually make these drugs. Y'all do know this, right? We don't make this. We don't know how to make this. Who makes this? Penicillin, methicillin, what? Nature, fungi, right? We didn't come up with that. All we can do is take this and add stuff to it and modify it a little bit. We didn't come up with any of that crap. Yeah. So we are well behind the times in science for not being able to develop antibiotics on our own. This is an area that we need to focus on, because you're going to get freaked out before the end of class, I promise. All right, so if you give the bacteria methicillin, methicillin also binds to transpeptidase, but it doesn't bind in the active site. It binds someplace else, induces an allosteric change, and now transpeptidase is inactive, right? So even though the bacteria are producing beta lactamase, it doesn't have any effect on methicillin, right? So now you can get mutations in the transpeptidase that prevent binding of the methicillin to the transpeptidase. And it can come about naturally through natural selection from mutations, or you could do that same DNA exchange. But either way, you get a transpeptidase variant that will no longer bind methicillin. So now you have MRSA. Most of our antibiotics are just modifications of these two major classes. So if you have something that is methicillin resistant and penicillin resistant, and in order to be defined as MRSA, you have to, number one, be penicillin resistant, and number two, be methicillin resistant. That's not said, but it's true. You've already eliminated a huge range of antibiotics. And so we're running out of things to go to after that, because things are even becoming resistant after that. Okay, So I want to show you this. And I put this, so in case you want to share this with somebody else. Where did I put it? Okay. Where? I put it this morning. Oh, yeah. But I put another one though, but that's okay. Okay, so this, is this it? Yeah, this is it. Okay, wait, dismiss. Okay, so this is so insanely cool. So this is an experiment that was done at Harvard. And they're going to take you through the whole experiment and explain exactly what happens. And then we're going to talk about it after, okay? So just sit back and try not to get too freaked out. It's really cool.
So we ended up building a basic that is 2 feet by 4 feet. So we set up that there are 9 bands. At the base of each of these bands, we put the normal petri dish they've had with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band is 1,000 times as much antibiotic. And then across the top of it, of course, thin agar that bacteria can move around in. The background is black because there's ink in it, and bacteria appear as white. First, you see they spread in the area where there's no antibiotic up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, spreads until it starts to compete with other mutants around it.
Just wait. Just wait.
When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
Okay, I want to back it up and look at the picture. What do y'all think about that? Isn't it though? Isn't it though?
Successive mutations
Okay.
that
So this, this is not bacteria, right? This is a yeast or a fungi, something, right? It's just contamination. But what you can see is, I love this picture because what they're doing is they're tracking, right? This was the original cell that developed a mutation, and then they track it, and I just think that is so cool. And then you can actually pull bacteria from different points and sequence their DNA and look at what mutations have facilitated their resistance to that much antibiotic. But how long was that? 11 days. Only 11 days. Isn't that kind of creepy? A little bit. So this is what I thought was going to be the next pandemic. Maybe still will be. We'll see. But it's something really you need to worry about, especially in the medical field. If you are in a hospital setting, right? You don't just have regular people in a hospital setting. You have immunocompromised people, right? So they're even more susceptible to infection. And then you have people who are infected coming into the hospital. I mean, it's just it's just so much going on in terms of trying to keep everybody safe and healthy and all that kind of stuff. All right, pretty cool, right? You want to go wash your hands, don't you? Even more than before. Yeah, this is why one of Charles's first words was hand sanitizer. It actually was. And I think I forgot to tell y'all a joke today. Okay, so this one, okay, wait, I gotta remember, because he pulled one and I knew everybody would know it. What do you call cheese that's not yours? Nacho cheese. Everybody knows that one. We pulled another one. Why did the teacher wear sunglasses during the day? because her students were so bright. Okay. All right, so I bring this slide back up again, right? Because now we're switching gears from bacteria to eukaryotes, right? Okay, so I just want to make sure that we're following along what organism we're in and where these different things are, right? Okay, so glycoconjugates have a huge role in cell signaling, in immunity, in host pathogen interactions, all kinds of things like that. So we kind of defined the interaction as either being intrinsic or extrinsic. So intrinsic, there are two kind of ways. I'm going to show you the picture and then I'll come back to the slide. But intrinsic recognition, you can actually have a host cell that expresses a glycosylated anything, right? Whether this is a protein or a lipid, doesn't matter. And there is something that binds to that glycosylated lipid or protein or whatever it is. Now, we call these things that bind, we call them primarily lectins. That's not the only type of glycogen binding protein, but lectins is a huge portion of it. So it can be within one cell, or it can be within two host cells. So let's talk about some examples between two host cells. And it doesn't matter where the glycoconjugate is versus where the receptor binding protein is, right? It could be one host or the other, it doesn't make a difference. But for example, if you get a splinter in your finger, right? There's probably bacteria on that splinter. So why don't you have this massive infection every time you have a splinter? You pull it out and wash it, right? But probably some bacteria is still in there. So why? You have an immune response, right? Okay, so what kind of immune response? You actually have to have cells that will go in and engulf and eat those bacteria, right? But they are not typically floating around in the tissue of your finger, right? Where are your immune cells? In the bloodstream. So how do you have cells that migrate from the bloodstream into the tissues? Well, it's kind of a cascade of events, right? You actually have injured cells, right? They release what are called cytokines. Cytokines are secreted molecules that are like the alarm system. And they say, I'm injured, I'm hurt, maybe there's some bacteria over here, please immune system, come here. And so the white blood cells that are normally circulating in your bloodstream hear the alarm and they enter the tissue. But golly, they go from floating around in your bloodstream to entering tissue. How does that happen? Well, it's kind of multi-stage. Let's say that this is a cell that's in a vein, right? And this is your white blood cell. Typically, the white blood cell is not expressing particular glycoconjugates that are required for the interaction between a white blood cell and an epithelial cell on a blood vessel. So the immune cell has to upregulate the little glycoconjugates that are out there, right? So now they express them. Now they can catch on to the vein epithelial cells glycoconjugates that are sitting on the outside of the cell. So basically what happens is you have these white blood cells that are like flying through the bloodstream. And when it starts to recognize cytokines, what they do is they express these glycoconjugates, and you'll see them actually start to slow their roll. And they actually they start out fast and then they tumble slower and slower and slower until they get stopped. Then once they're stopped, that means they're making the maximum number of connections. They actually squeeze between epithelial cells of veins and enter the tissue. So I want to show you an actual video of the white blood cells doing this in the bloodstream because it's so cool. You want to see it, right? Here. Oh, I put it in the wrong place. I got to move that up, but it's right there. Okay. And we're not going to go the whole video. There's a little animation. And here we go. Okay. This is what I want to show you.
Leukocyte rolling is observed directly in an anesthetized mouse. The up and down movement of the frame is due to the mouse's breathing. Two blood vessels are shown. The upper one is an artery with blood flowing from right to left. The lower one is a vein with blood flowing from left to right. Leukocytes only adhere to the surface of veins. They do not crawl out of arteries. Some leukocytes are firmly attached and are in the process of crawling through the vessel walls, whereas others have already left the vessel and are seen in the surrounding connective tissue. When the blood flow is stopped temporarily by gently clamping the vessels, we can appreciate how densely both vessels are filled with red blood cells. Red blood cells do not interact with the vessel walls and move so fast on the normal flow that we cannot see them. When the blood flow is restored, some of the leukocytes continue rolling, whereas all non-interacting cells are immediately washed away by the shear.
Not really cool. So depending on what is on the surface of the cell tells you if it's going to stick or not, right? Red blood cells are not going to have those glycoconjugates sitting on the outside of their cells. You know, I don't know what we're going to be watching next. All right. So that's kind of host-host interactions, right? But bacteria, viruses have all co-evolved over a very, very long time. And so basically what they do is they hijack that system and they target particular cells. So they mimic a host cell. So they could mimic the glycosylated protein or lipid or whatever it is so that the cell will bind to it, or they can mimic what will bind to the glycosylated protein or lipid, right? So they can go either way. But it happens with all kinds of different pathogens. But this is our extrinsic recognition, right? Where we're looking at something outside of the host. So let's look at a little bit more examples, right? So what I showed you was this. This is our immune cell, right? This is like the white blood cell. And so this would, for example, be the epithelial cell in the vein. Now, why a vein and not an artery? Anybody know? Arteries are too high pressure and their walls are super thick because they have to be really thick to withstand the pressure. Veins, on the other hand, are thin and lower pressure, so it's just easier. Okay, so this would be the vein epithelial cell. This would be the white blood cell. And you would have many, many, many of these lectins that are there to bind the glycosylated proteins or lipids or whatever it is, right? Now, there's a particular kind of E. coli, and the E. coli have a protein called FimH. FimH is a lectin, right? So it will bind to a glycan group on a protein, and the protein is uroplankton. Now, why is this important? Do you remember when I was talking to y'all about mother's milk and that there were sugars in mother's milk that mimic what's on the cell? So like, why is that important? So this E. coli is the E. coli that is primarily causes urinary tract infections. So that's how this E. coli will hang on, right? So when you urinate, right, they have to kind of clutch onto something so they don't get washed away. So they'll actually bind, and then they have little flagella, and when you're done peeing, they'll swim up your urinary tract, right? So that's how they survive. But what will actually happen in mother's milk is mother's milk has a glyco mimic of the sugar moieties that are attached to the normal protein, uroplankton protein. And so it will actually prevent a UTI from happening because it mimics it and it soaks up the bacteria, and therefore the bacteria can't bind. And it's a protective thing. So it's really cool. It's really cool how everything's so interconnected. Okay, and that's true for, right, bacteria, viruses, all kind of.
It's similar.
Right, so this is like the individual glucose moiety that's right there. Right, so this is a zoom in of this. Here's our E. coli. The E. coli have a FMH protein that's in green. This is green, that attach to a sugar moiety. So basically, mother's milk has a short a short carbohydrate chain that mimics just the end. So it soaks up the binding sites from the bacteria. So it can't bind to cells, right? Because the bacteria can't tell the difference. They're not smart. They're not smart. They are evolved, but they are not smart, right? Okay, so how do the linkages occur, right? We've talked about all these peptidoglycans and proteoglycans, and so how does that actually happen? How do you get sugar to attach to, for example, a protein? So there are two different ways that you could do this. You could do this through an N linkage, or you can do this through an O linkage. So think about the structure of the proteins, right? You know your proteins, you know your amino acids. So what is ASN? Asparagine. Asparagine has a ring with a nitrogen. So this is why we call it N-linked, because your sugar molecule will bind to the amino acid in the protein at the nitrogen. So this happens only in asparagine, and it actually happens not just asparagine, but it's like asparagine attached to particular amino acids, right? It's not every single asparagine that it will actually bind to. There's actually a recognition site, but the linkage occurs to the asparagine. That makes sense? Sort of? Yeah, so it's like a recognition site and a binding site. They're two different things. Where the bond forms and what is recognized by the enzyme in order to make that linkage occur. On the other hand, you can have an O-linked, right? Most sugars are O-linked, right? If you're just doing a sugar chain, it's O-linkages. So where do you have oxygen? Serine, and it's primarily serine and threonine where the O linkages occur. And you have usually a hydroxyl, right, an OH, and that's where your sugar is actually going to bind. And so you can see this is an example of an acetylgalactosamine, right? And then you can go on and build whatever other sugar structure depending on what glycoprotein it is, right? But now, would the same enzyme glycosylate asparagine as serine? Absolutely not. The recognition sites for anything involving sugars is super, super specific. So it's specific when you glycosylate the protein, then it's specific when you continue the glycosylation on top of it, right? So if you're adding the next sugar moiety, it's a specific enzyme that's going. So why is that important? Well, it's super important for ABO blood groups, right? So our ABO blood groups are a combination of glycoproteins and glycolipids. And we're really just going to talk about the glycoproteins. So if we look at someone who is a blood type O, you've learned about blood types? Yeah, good. Okay. So a lot of times we think about O as not having anything, right? But that's not quite true, right? Every time I teach you something, I'm like, well, I kind of lied a little bit. So every ABO antigen, right? That's the stuff that's sitting on the outside. right? The antigen is what sits on the red blood cell. So every single antigen, whether it's O, A, or B, has a core that is exactly the same. So it's a set of 4 different sugars that are attached to either a protein or a lipid. Now, people who are type O, this is all they have, right? If you are type A, you contain an enzyme called the GTA enzyme that will actually take an N-acetylgalactosamine and add it to the O antigen. So now I have an O antigen that's in the orange, the yellowish square, but I added an extra sugar moiety to it, right? Now, if you are a type B, you have the enzyme GTB. I always want to say GTP, but it's GTB. That transfers, what is GAL? Just plain old galactose, right? This is N-acetyl galactosamine. This is galactose. So this will add a galactose to the B type, to the O, to give you a B type. Okay? So does this explain why you can have people who are AB? Absolutely, right? What if you contain both enzymes? If you contain GTA and GTB, you are AB blood type, right? Yes. These yes, but delta G is.
So we can't like make blood that's AB. So like.
Oh no, I mean like you have like millions of these on the outside of the red blood cells. If you miss a few, you're gonna have an immune reaction. So like that just sounds insanely dangerous to me. maybe you could take red blood cells and one day in the future grow them in the lab and just mutate the DNA so that you have whichever active enzymes. You take somebody who's AB, right? And then you want to make A, you mutate this enzyme. You want to make B, you mutate this enzyme. That's possible. Well, maybe. You know what I mean?
I mean, with CRISPR technology, they could add mutations into the
Yeah.
DNA that
I mean, yeah. But I wouldn't take blood that was already there and then enzymatically treat it just because I'm afraid you would miss some. You know, but the idea is right. If you were able to remove just that one, then you would revert back to O. Absolutely.
will just
Keep what?
someone like a blood type O. They can get spot, they can do that, they can do that.
Well, if you're O, you don't have either enzyme. So you'd have to start with somebody who had both if you wanted to make just one.
It only adds to the yellow circle.
Correct. It only adds in one particular place, right? Because the enzymes are super insanely specific, not only for what they add, but where they add it.
And then do we have to know the full name of GTA or is it actually called?
Yeah, what is GTA? What does it stand for?
Transferring.
Well, it depends on which one it is, right? So if you're transferring N-acetylgalactosamine, so this is N-acetylgalactosamine transferase, right? But they just call it GTA. Here, this is GTB, so this is galactose transferase, right? Yes, it is. Yes, it is. You have to add it to a galactose. Yes. Because these enzymes will only add to galactose. Yeah. It's pretty neat.
But the difference between GTA and GTB is the N-acetylgalactosamine or the galactose.
Correct. Is the ability, it's not the enzyme. The enzyme doesn't have the sugar. Be careful with that though. The enzyme doesn't have the sugar. The enzyme has the capability of adding that particular sugar, if that makes sense. Yeah. Okay. So, right, and you'll know all your compatibility stuff. You have to really, anybody not a blood type person? Right, so if your blood type, I always hate these big diagrams, right? So if you are, let's say you are type A, right? That means here's your red blood cell. You have A antigen sitting on the outside of it, right? So can you receive blood from somebody who is AB? Can I give blood this way? I cannot. I cannot. Because whoever is type A, this is your antigen. You have antibodies that are not anything that's not you. So you have anti-B. If you had anti-A, you would clot your own blood, right? So you have anti-B. So if I gave a red blood cell that had A and B to this person, I have anti-B and I have B, the blood would clot. Yeah, you have it's an agglutination reaction. Yeah. So you have to think about what type you are, what type of antigens you would have, and therefore what kind of antibodies you would have to that reaction. You want to do another one? So for type A, let's write this. Can receive, this is my trick. You can receive anything that is U or less than U. So U is what? Type A. What is less than type A? Type O. Now we're not talking about RH factor, right? Like we're just not going there today. We're not going there. We're not going there. Okay. So now who can you give to? Type A can give to, can donate to. You can always donate to yourself, right? So to you or more than you. So if I am type A, I can donate to type A, and I can donate to type AB. If you remember those two statements, you're good. You don't have to memorize a massive table. That's it. They do. Mm-hmm. Yep. Anybody want to do any more? Are we good on that? I'm happy to do any comparison. What's that? Okay, so if you're type O, let's do this. Type O. Type O. Type O. It's a typo. All right, so type O, we want to know who we can donate to, D-O-N-A-T, donate, and who we can receive from. Donate to, receive from, okay? So if whatever type you are, you can receive from you and less than you. So you can receive from you, which is type O. What's less than type O? Nothing. Okay, so who can you donate to? Yourself. Just type O. Oh, everybody. Type A, type B, type AB. Right. That's why they call type O the universal donor. What's that? Just O negative. Yes, just O negative. Yes. Lots of people have O positive. Not so many have O negative. O negative is the universal donor. What's the universal recipient? AB. AB positive, right? If you're gonna do it, AB positive. If you're gonna do it. So like, yeah. A negative is like type O. A positive is like either A or B. It's like present or not. It's not that there are variations like A and B. It's just like there or not. That's why they call it plus and minus, there or not. That's what the RH factor is. Yep. Yeah. So if anybody wants to go through that, they can come see me. I'm happy to do that with them. All right. So I want to give you something. I think, where are we? I don't think we'll be able to get to both of them. So there's one on the front and one on the back. So I want to do the lectin one first. So what's a lectin? It binds to glycosylated, usually proteins, right? So anybody heard of ricin? Yeah? So who typically gets ricin in the mail? Presidents. Lots of presidents have gotten mail packages with ricin in them, right? It's very, very dangerous. So what I have here is just a little paragraph for you to read, and then we're going to talk about basically how these lectins are interacting. So there are a few questions you might need the internet for. So, but we'll talk about it. So I want y'all to work. You can work together. You can work alone. I don't care what you want to do, but we'll come back together. Just do the front page. If you're done early, you can start on the back, but do the one with ricin, the world's most dangerous lectin, first, and we'll see how that goes.
Yeah.
Hmm?
I've heard rice.
You've heard what?
Yeah, I've heard rice.
Yeah. Yeah, now I'm going to tell all you guys to Google it, and now we're going to be on like the FBI list or something. Right, exactly.
Let's be honest, we're not gonna list the back.
Well, you know, that's chemist. That's what it is. Chemist. Okay, I'm going to pause this. Okay. So how does ricin gain access to the cell interior / cytoplasm? Well, that's how it gets in the body, but how does it enter a cell?
The linkage to the
So either it binds to N-acetylglucosamine or galactose, right? Either way, but that says it binds. So then what happens? Anybody look into that? Like, okay, you're gonna have this lectin that's gonna bind to a glycosylated substrate. And what happens? It what? It goes through a particular kind of endocytosis, right? So it's gonna bind like this, right? And then what actually happens is the membrane kind of divots in, right? And you still have that bound. And then eventually the two sides will meet, right? And then this part of the membrane gets pinched off and it has the lectin on the inside of it, right? Okay, are we in the cytoplasm? Cytosol, cytoplasm. We're still not, we're in a vesicle. We're in a vesicle. We got to get out of the vesicle. Where are we going to go to get out of the vesicle? We're gonna do retrograde transport to the Golgi. It'll hit the Golgi, right? And then it'll dump the contents into the cytosol, cytoplasm, whatever you want to call it. Right? It's retrograde to the Golgi. Yeah. Okay. So that's how it gets in, right? Okay, so now, is it in its dangerous form?
Yeah, when it gets in the stuff, when you've already ingested it.
You've ingested it, right? Okay, so not yet. It's actually not yet, right? Because it's actually composed of an A chain and a B chain, right? And when the two are linked, it's fine. So what happens? In the ER lumen, it goes to the ER lumen, A cleaved from B. Now we got problems, right? Which one is going to cause problems? The B chain is what got us in. The A chain is what is going to bind to ribosomes. Did you see this? A single ricin molecule will inactivate, brace yourselves, 1,500 ribosomes per minute. Per minute. Y'all, that's insane. That's insane. Okay. So why does a ricin kill the castor bean seeds?
Stored it in protein body, and it's not catalytically active until they get the AMP chain get weak.
So it's stored, right? That's one thing. It's stored. And then it's also in a proto form. Do y'all remember that from Biochem 1? Where you had something that is, you know, a whole protein. And then in order to make that protein active, you have to cleave it. So it has a proto form, proto-ricin form. Right, that's exactly right. Okay, so what potential ways could ricin's activity be used in human medicine? List as many applications as you can. What? 2? Pro-ricin, R-I-C-I-N, that's supposed to be a C. So the A and B form are together. When the A and B form are together, it's fine. It's only when it becomes cleaved that your A form is able to go and inhibit ribosome. So if you're in the pro-ricin form, it's cool. A and B are together, it's all good. No, as long as they're sequestered and stored in the right place. Right? So it's got to be twofold. That's not just a one-part answer, right? That's a twofold answer. Okay, so what potential ways could we use this activity as a plus? So it's toxic. So we could use it to treat any cells that we want to get rid of. Cancer cells included, sure, absolutely. But if you ingest it, if you inhale it, you will die in 3 to 5 days.
don't know that it's gonna target those functions.
Okay, so the problem is we don't know where it's going to go. It's going to go ubiquitously everywhere unless we tell it where to go. How do you tell it where to go?
Or putting
Target to, we'll say any cell we don't want, but we'll use cancer as an example. How do you target?
Find some way to- You have to find a receptor on the cancer cells that's not on the normal cells, or mutate the cancer cells more so where they express a certain receptor that's not on normal cells.
So cancer cells are really cool because they do express things they're not supposed to. And cancer is a unique disorder because it is so insanely unique. You know, like if everyone in this room, knock on wood, if everyone in this room had cancer, right? And even if we all had the same form of cancer, like leukemia or whatever it is, knock on wood again, right? Okay.
We're really putting some bad
If you
vibes.
isolated, I know it's just a bad day, right? If you isolated all of our cancer cells, each one would look different. Because it accumulates, to get cancer, you can accumulate mutations in different genes, right? So everybody can have a unique combination. So everybody's cancer is not going to respond the same way to the same drug, right? So the new thing, right, would be to take out cancer cells and then conjugate the ricin to an antibody that would specifically bind to your cancer cells. So when my husband was doing his postdoc, he was actually doing cancer therapeutics, not with ricin, but with other molecules conjugated to small biomolecules, not necessarily antibodies, but small biomolecules that would specifically target, like they would give a mouse cancer, then they would take cancer cells out of the mouse, culture it with these different little biomolecules, find out which ones would bind the most, then conjugate those biomolecules to cancer therapy like chemotherapy and give it to the mouse, back to the mouse, right? So it's in the works. Like the future of cancer therapy is individualized cancer treatment. It's really cool.
Like they take so personalized.
Personalized, but it has to be. It has to be.
Could you also use castor beans from the ricin to genetically modify plant organisms to try get rid of certain.
Well, you could also put the gene that codes for ricin under control of a particular transcriptional regulator, a particular promoter, all that kind of stuff, right? But it would be expressed only in a particular cell type. Like if I want to make,
Do it in order to combat pesticides and herbicides that are hurting the soil. This is a more natural way, I guess.
well, so if you're gonna do that, we're actually gonna talk about that later in the semester. If you're gonna do that, you actually want to create plants that are resistant to something that you spray. So then natural plants die like weeds, and your genetically modified crops survive.
That hurts the earth more than helps it.
Yeah, but the way you said it doesn't work. Any questions? If you're done, you're good. You could go if you still want to chit chat. You can stay and chit chat.
Go back to your, right, like, instead of antibiotics.
It's not, because this is the problem, right? If you have a bacteria and I give you ricin, you're gonna die too. That'd be cool. That'd be cool. So in the future of fighting bacteria might not be antibiotics.
I mean, it's gotta be something else, because I mean.
That's the way you go.
So when y'all get Nobel Prizes for, like discovering some new molecule that will have the properties of antibiotics, not be an antibiotic, like all I want is like a little shout out, just a little shout out.
Tiny little
Just
shout
a
out.
tiny-
Thank you
Tell you
to
what.
my professor.
To my Nobel Prize.
She heard me
Just a
with the
tiny
rice
shout out,
and
not
the
another
I'll say, I would like to
class.
thank
My biochem professor slash possible ethical terror.
for getting me on this path of discovery
Absolutely.
to revolutionary molecules that will change cancer forever. I mean, there's also like xenobots as well.
Oh, yes. Absolutely, absolutely. There's not one way to attack the problem.
They're all really far back.
Cool, whatever. Okay, so I'm gonna end the Zoom.